Greetings to IPC 2021 participants. I'm Gleyci Moser, Associate Professor from Rio de Janeiro State University. Our study entitled Comparative Characterization of Tetraselms chuii, Chaetoceros calcitrans, and Isochrysis galbana strains as source of food supplements is part of Ecosplora project. The species of the genera Tetraselms, Isochrysis, and Chaetoceros have been traditionally used as valuable nutritional source in aquaculture for their high fat acids content. In recent years, several microalgae have been incorporated also in food products. Here we evaluate the potential of 5 microalgae strains to produce polysaturate fat acids, Profª, with interest for their use as nutraceuticals. Cells were cultivated in F2 media, scaled up in 12-liter tubular photobioreactors and maintained at 20 degrees centigrade and constant PAR. Growth was monitored by cell counting using an image stream cytometry (AMINIS) that enabled us to measure morphometric parameters. Samples were collected, concentrated, lyophilized, and immediately frozen for analysis of Profª during exponential growth and late exponential growth. Profª were extracted and analyzed in a GCMS using high resolution. The graphic shows the variations in biomass, total fat acids, and Profvä's during exponential and late exponential growth phase, comparing the 5 strains considering both surface volume ratio and maximum linear dimension. The biomass from the 3 strains of Tetraselms chuii were greater than those of the other strains tested. Total fat acid ranged from a minimum of 9.3% to 32.18% of the total dry weight. All strains varied its Profväcontent with culture age. Yield by cell, fat acids and Profväwas 10 times higher in Tetraselms chuii strains than in Isochrysis galbana and Chaetoceros calcitrans. TCBG1 strains showed higher biomass and Profväcontent during late exponential growth phase, while TCBG2 showed higher biomass and Profvä during the exponential growth phase. All strains produced significant quantities of specific omega-3 acids with interest as nutraceuticals, and Profväa production ranged from 36 to 71% of total fat acids. Particularly, strain TCBG1 produced arachidonic acid and EPA at the exponential growth phase, while TCBG2 produced DHA at exponential and EPA at the late exponential growth phase. A two-stage growth system using co-cultures of 2 Brazilian Tetraselms chuii strains in photobioreactor is proposed as an optimal model for Profväa production. Thanks for your attention.